just sort of
just start.
You can introduce yourself and just start or whatever.
Okay.
Well, this is Heather Mefford.
For those of you who don't know me.
And I'm Barb Trask, and I really appreciate being included in this illustrious lineup for this series.
All right, so I think we'll just get started with the early years.
Okay.
Tell us a little bit about where you grew up and how you got interested in science.
Okay. I'm the oldest of 4 kids. Grew up in Michigan and outside of Chicago, or in reverse order, outside of Chicago and then Michigan. My parents are college educated. If I think back of how I got interested in science, it has to have started in the museums in Chicago, where we went regularly to the Science and Industry Museum and the Natural History Museum. And I think I was probably one of these kids that is so irritating to the rest of the family because I wanted to read every word of every sign on every display. But the real cementing of science was a high school biology teacher who was very unorthodox. It wasn't the standard kind of biology. We dissected cats that had been brought in with latex-filled veins, and he took us on field trips, and it was just eye-opening. He did oral exams rather than multiple choice, and it was very exciting. So I think that was what got me really launched into science.
Great. And so tell us a little bit then about your college training and graduate school and how you got to that point.
Well, I went to school at Purdue to be a good distance from my parents in Michigan, and I went thinking that I was going to be a doctor or something like that, and I'm glad I didn't. I ended up migrating to wildlife ecology, so my degree's in wildlife ecology. I got into a project, so then you got to think about how did you get into research rather than just being interested in biology. And I think that came by a guy whose name is Jim Carr, who ended up here actually in fisheries department many years after, who got me involved in a fisheries project dissecting fish, and I must have gotten a lot of formaldehyde all over me in that period. And was the guy that said, why don't you apply for an NSF fellowship? You can do this. Mm-hmm. And he ended up leaving, and I ended up staying and working on pheromones in Syrian hamster and a variety of things. And then I met Ger, who many of you know is my husband, and I ended up in Holland, where I did research. Ger had gotten the very first flow cytometer in one of the first ones in Europe, and we ended up working together on trying to sort mouse hematopoietic stem cells, and I kept trying to migrate back to aquatic ecology and had a project that was funded by the Dutch organization that takes care of waterways. We ended up separating or distinguishing different species of algae using flow cytometer, and it was my first paper, and the first application of flow cytometry in that arena. And now they got flow cytometers going on ships and measuring things that people can't see through the microscope. It was kind of a fun time to get going.
And so you spent your doctoral training was mostly in Holland? Then?
Well, at that time I was just sort of freeloading and working in a lab, not getting paid because I was working in Ger's lab, and we felt that it was inappropriate for me to be taking a paid position when there were a lot of unemployed people in Holland. And so I was just working, and we lived out in the country and had a farm, a little teeny farmlet. So no, I didn't get my degree until after Ger had got money. He had an appointment, and he got money for a sabbatical that took us to Los Alamos and then Livermore, and in the time that the Genome Project was just getting off the ground. And we went there to try to push the technology of flow cytometry more by applying it to chromosome characterization and sorting. And at that time, that was a key technology in the early phases of the Genome Project. It's hard to believe when you think now that you can just take a whole tumor and dump it in the sequencing hopper, or a whole sample out of a ecosystem, that we actually had to first try to build a map and parse the genome up into small overlapping bits to assemble it. And one of the strategic steps in that was to sort chromosomes to simplify the problem. So we got involved in that, but went back after this sabbatical year to Holland in the hopes that we could sort of, I don't know, we naively thought that Holland would pick a chromosome and we would be and we would be involved in some way in working on it. That was met with a lot of skepticism because we weren't human geneticists. We didn't, what did we know? And so we started thinking, well, this is not going to fly. And at the same time, Joe Gray, who was one of the key players at Livermore in the Genome Project and flow sorting and that sort of thing, was trying to get us to come back because he felt that we had some skills that were useful. So what I did was, in that year and a half when I was back in Holland, I got the stuff together for my thesis. The Dutch have a strange system. You can actually leave college and then accumulate a body of work that you then go back and say, this, I think, is worth a PhD. And so that's what I tried to do. And that was the year that I got to learn about fluorescence in situ hybridization, or FISH, because that was being developed in a lab in Holland, some of the early stages. And so my thesis was about using FISH and flow cytometry together to try to quantify aneuploidy in nuclei by the fluorescence intensity of bound probes. So it was sort of the merging of these two things.
And so really it was those two technologies then that you came back with in your kind of, as you started your career before you came to the University of Washington, right?
Right.
And so tell us a little bit about how you used those, as you already mentioned, in the early phases of the Human Genome Project, and what types of problems you were working on within the context of that project and your own other research.
Yeah. That's a good question. Those two technologies were used for a variety of projects that had in genomics and human genetics. The first was trying to sort chromosome-specific DNA that libraries could be made of, and then those would be then pawed through to try to make an overlapping map. So I was part of the production phase of that. We had to grow up hybrid cell lines because you didn't want the chance of contamination of other chromosomes. So there was a lot of characterization of the source material and big farms of cells that you could grow up enough cells to pump through the highest speed sorter at the time. And Gare was involved in the sorting side of things, and he went on to continue to develop flow cytometry as a tool for cell biology. And I sort of migrated more towards the human genetics side of things, applying both FISH and flow to look at chromosome abnormalities in human genetics problems, looking at normal human variation, like you were involved in the flow cytometer can detect big differences in the DNA content of chromosomes from one individual to another. So it's sort of the early phases of structural variation that you could actually both see under the microscope and quantify in flow.
Both normal and disease.
Yeah, and as an example of a fun project where FISH factored in was actually looking through the microscope at cells of individuals with Charcot-Marie tooth. This was a collaboration with Jim Lupsky and Pragna Patel, and they'd done all this fancy genetics and fancy molecular biology to try to determine that there was an extra copy of a segment of chromosome that was causative, and we could just use a probe for it, look through the microscope and see 3 spots. And it was just, to have this technology where you could see see features of the genome that had been invisible or through other techniques.
Yeah, really exciting.
Yeah, that was fun.
So then tell us then kind of how you and Gare both actually migrated to the university and what was happening at the time.
Yeah. The we stayed in Livermore for 7-8 years, and it was an interesting time because it's that when we first went there, it was a happening place. There was lots of radiation biology and cytogenetics and the early phase of the Genome Project. But later on, it became the Genome Project actually narrowed the focus of the place. And so we were anxious to leave and get to a place where there was more diverse science going on and in an academic setting. So we started contacting people and saying, we'd like to work elsewhere. And we started with Lee Hood, actually, when he was still at Caltech, and said, you need somebody like us. And then there was a point where he was going to maybe set up shop in Berkeley, and we approached him again. I remember just sort of tagging behind him at meetings and trying to squeeze in where he's in the line and remind him that he was missing these two technologies and our approaches to science. And when he got arranged to start molecular biotechnology here, we were on his case again, and he let us in. so in fact, I was thinking back at that time, and I remember when we met him up here, and he was getting ready to do his spiel to make an offer and say, and we just cut him off and said, just save your breath. We're going to come. Don't. And he said, well, I've got to do this. I have to tell you all these details. So he had already given out all the state line positions, so all the regular faculty positions. So Gare and I both left what were essentially tenured staff scientist positions at Lawrence Livermore and took research faculty positions. And luckily we had good grants that we could bring with us and support ourselves. And he did come good on the promise to, when new state line positions came available to the department, that we would get them, and we ultimately did. So it was fun. Yeah.
So I wasn't here quite at the beginning. I've been here a long time, but I can imagine it was quite an exciting atmosphere as far as a new department and a fusion of technologies and new students trying to get in. Can you tell us a little bit about the atmosphere of MBT?
Well, you actually were in the early phases. Heather worked as a tech in Gare's lab, and she was involved in analyzing chromosomes and looking for polymorphic variants, and had a nice paper. And that was in Fluke Hall, where we were all compressed together. I was sharing an office with Maynard, and my half bench was backed up to Debbie's. But it was
kind of nice to have everybody interacting actually in a big open space.
It was great.
Open space.
No, it was one. In fact, it was a very small footprint. It was fun. No, it was I thought it was just a really exciting time because of the I don't know, a lot of energy and a lot of excitement about interdisciplinary research. There were faculty that were excited to be thrown in with faculty that didn't actually even know what they did. I remember trying to just concentrate on what John Yates, who does proteomics, what he was trying to say, and see, I've got to be able to understand this, And there were people coming to the department from across the university to learn more about how they could be involved in the genome project or in biology, especially from computer science and the
engineering. Right?
Engineering. There was students who were just coming from non-biological backgrounds and excited about having a degree in this arena that was at the interface of technology and biology. So they were excited. There were students that had read about it someplace and they said, I want to go there, and that's it. So it was and the faculty were excited about trying to teach biology to engineers and computer science, and have the biologists learn more about computer science and math and physics. And so it was really exciting time.
So it must have been a little bit challenging to have such a diverse array of both students and faculty. Were there challenges that came with that, or was it more an environment of just kind of mutual learning and excitement?
Well, if I look back on it, it may be that the parts were almost too divergent. I think that you had one of this and one of this and one of this, and so it's hard to get some synergy going. But I still remember it as a pretty exciting time.
Yeah. I have to check my cheat sheet. So I guess once you got here, in those years that you were with molecular biotechnology, what were the problems that your lab was working on?
Yeah, well, so I was continuing to use, we're still using flow cytometry a lot for human genetics projects, sorting chromosomes that had rearrangements so that molecular biologists could map breakpoints and define deletions and things like that. But increasingly, my work was focused on fluorescence in situ hybridization and using that as a tool for a variety of things. One, for example, is trying to look at how the nucleus is organized by measuring the distance between points that you know how far apart they are on the DNA molecule, but now you're asking how are they situated with respect to each other in the nucleus. And this had we'd gotten into this because we realized that you could use distance as a way to help map the genome. So if you had two sequences and you were trying to figure out how contigs were situated or whether you'd put a contig together correctly, well, if you took two probes and said, well, they should be close together. They should be close together in the chromosome. They should be close together in the nucleus. And then we realized when we started looking that you could actually use this distance as a map building tool. So it had this practical consequences, and we wanted to know what the limits were, but it also had the ability to probe how the DNA is folded in the nucleus.
Biology.
Yeah. So that was one avenue. Another avenue was the grew out of taking clones and just saying, how do we link the sequence or the growing sequence to the cytogenetic view of the world? There really wasn't a way of connecting the two. So you had to use FISH to say, I've got this piece of DNA and it's in this band. And so we were trying to link the cytogenetic map to the assembled genome sequence. And in some of those clones that we picked, even though we streaked them to single colony, they lit up more than one place in the genome. And some of them, like ones that derive from chromosome 19, hit 19, but they hit a bunch of other places. And so that launched a long interest in how what were these sequences that were duplicated, and how did they evolve? What did they contain? How did they move around? And the one that just got every student tired was this one that mapped to multiple ends of chromosomes, and we made at one point I don't know what possessed us, but we hybridized it to two different individuals, and the pattern was different in these two individuals. So we realized that we were looking at structural polymorphism, that here the ends of the chromosomes were different in normal humans, and then we started looking at lots of people and chimps and tried to figure out what these contain, and it just burgeoned.
So that really took you in a new direction, and I think was a significant contribution to the Human Genome Project and our understanding of really chromosome structure and variation.
Yeah, it was sort of the first, cytogeneticists had seen that there was gross differences between chromosomes of healthy individuals, but most of that was near centromeres and was composed of repetitive sequence. But this was out in the in sort of the normal turf of,
Correct.
of chromosomes, and if you sequence this piece of DNA, it looked, it had genes in it. It was so, Evan and other people have really uncovered a vast amount of variation of this type, but it was one of the early
Right.
ones. Yeah.
Yeah.
And it was only really possible using fish because we
Right.
could see it.
Mm-hmm.
Yeah.
Right. Yeah. Great.
And then that piece had olfactory receptor genes in it, so that got me interested in where are these genes and
Are they
how do they
functional?
work, and
Yeah.
that sort of thing.
Great. So I thought maybe we'd switch gears just slightly and talk a little bit about who some of your mentors and role models were kind of throughout your scientific career, and how that shaped where you went and what you did.
Well, they were all men, because there weren't a lot of women role models, but they were all, there were a lot of them, and they all had the same, the characteristic that they all thought I could do more than I thought I could. And they let me know that, and in some way. And so Gare is one. He was my harshest critic, I think. You were around. He wasn't shy about telling me that I was working on that was a stupid idea. That was not why do you want to work on that?
Right, though.
Why do you want to work on that project, or would have a much simpler explanation for something where I concocted a complicated model to explain some data. So, and also very tough on thinking about how to present your work. One of the early memories I have of our relationship was sitting at my first big talk at a big meeting, and sitting on the bed in a hotel room, and he's going through my slides and saying, well, what's the point you want to make with this? And what was the point you were? And they were flying, and I had them all. and then he took what was remained, and he just said, well, how about doing? And he just said, well, try that. So I do that now. I still do that to students, to try to throw up all the cards. But at the same time, he was one of the people who said, you can do this. He was the one that encouraged me to respond to the Hutch when they asked me to be a division director, and he said, well, you can do this. It can't be that hard. So he's one, of course, and Dick van Beckham, who was the head of the research institute we worked at in Holland, one of these guys that was just bold and tough and cared very much about how you plotted data, and had very little regard for authority. And he's the guy that I learned to try to think for myself, because he didn't suffer fools gladly. If you came in, he would look over his glasses at you and tap his pencil, so you had to get your point out really quick and get out of there. But at the same time, he's the guy that I learned that it was so important to know the names of everybody that worked. He knew everybody that worked at the institute. He knew the family stories of the janitor and the person who ran the stockroom. And so he was all about very strong science, but he wanted to make a place where people felt like they were part of the organization, no matter what they did in it. So he's one. I think Lee Hartwell, who gave me a chance to be a division director there. He's you guys know him as a great scientist and a very charming guy. But you may not have worked under. He was my boss, and as the president director of the Hutch, and he was just a low-key leader. But from him, I learned every decision that we had to make about policy changes or funding or hard problems precedent-setting changes that might go into effect. He was the one that always stopped and held up a candle and said, "Are we making this? Are we choosing the solution because it's easy and it'll get this problem off the desk, or, and out of our way, or are we choosing the solution that will, in the long run, make the institution a better place for science and for people to work here?" And he always made us look at questions and problems with that light. So I still try to do that. And then Mark Rodin, who was my colleague at the Hutch, who was always willing to entertain a question about how to do something. He's the kind of he created a culture where it wasn't you didn't have to be embarrassed if you didn't know how to solve something in a leadership role about people and trying to manage people and grants or negotiating, or if you didn't know how to do it, you could just say so, and he'd be willing to offer advice and wouldn't be upset if you didn't listen to him. so those are the kinds of people that I really regard.
Good. Well, now I know where the Edward Tufte books
came
from.
That's right.
One of the things that I remember the most, I think that I learned from working with you was the value of a good presentation. And I remember that's the first time I saw those Tufte books, and they're in my house now.
Non-data ink.
Yep. No non-data ink. That's right. Great. So let's talk a little bit then about your lab. Over the years, who were some of the members of your lab, either students or scientists who contributed?
Yeah. Well, it's interesting. Because I came here relatively late, I didn't have a lot of graduate students. But when I think about the people that I really admired most about during my career are graduate students, people like you and Lena Linardopoulou, who really fearless and motivated and curious and just dove in. So you two for sure. I had the great fortune to work with Janet Young, who's over the last she actually started in Lee Hood's lab and migrated to mine before we moved to the Hutch, and she is one of these people who's not afraid to tell you when you're wrong, or that. So she's a great writer and a colleague. Had great postdocs that have worked with me. My last graduate student is Kyle Siebenthal, who's everybody's been so different. It's just and he's got this fierce independence and focus, and it's scary sometimes, but he's also, I think, one of the people that I'll look back on and be proud of his time in my lab.
So I think one of the things we skipped over that we were going to talk about was kind of that you've already mentioned the Hutch, but there was a period of transition for both you and I think the Department of Molecular Biotechnology, where Lee Hood had left to start the ISB, and you took over the role of acting department chair.
Right.
Can you tell us a little bit about that role and that time in molecular biotechnology?
Yeah. Well, it was tumultuous. That's about the best word I can give to it. Yeah, Lee had left with several people, including my husband Gare and Rudy, and faculty were up in arms about the department unraveling. Students were freaked out because they weren't sure whether their degree that they had or they were in the process of getting was going to have any value. If the department doesn't exist, what will my degree mean? There was we were faced with dealing with layoffs of 100 some people who had been part of a off-campus genome center that Lee had run, and that didn't have any money, and that was my first job, is to figure out how to gracefully help these people be laid off. That's something you don't want to do. and the university was saying that people were upset about it at the university because these students were going to come back into the pool and be sort of, you would have to,
right?
you would have to hire them too. There was not sort of, so it left people with little choice of whom they hired as well. So a lot of things going on. I asked the dean to come and reassure the department that everything was going to be fine. And at the faculty meeting, he really offered little reassurance.
What did he say?
You may not want to include this. He said that molecular biotechnology had not lived up to its promise, and the evidence for that was that his nephew, who was a medical student, had not heard of the department yet. and so anyway, it was a tough period, and that didn't help. The dean's office basically said to me, they didn't give me any guidance on how to be a department chair. They just said sink or swim. I think they were, kind of hoping that it would unravel under my leadership. So I felt like my job was to just keep the boat afloat and calm everybody, because people were freaked out, and just try to help people just stay focused on science, and everything's going to be okay. So in the middle of all this, that's when Lee Hartwell came and asked me if I would be interested in considering to be one of the division heads. There are 4 divisions there, so it was a real step up as well. And I had always had enormous regard for the Hutch and the institution from the time in Holland, because the institute in Holland was in the early 70s was also working on bone marrow transplantation. So there was you would think that I wouldn't have known about the Hutch, but I did. And so I was really excited to be. So when I told the dean that I was very interested in pursuing that opportunity, he wisely asked Maynard to be the acting chair, so I wouldn't be in a lame duck position. And then I went on to accept the position. But I think really the change, the dramatic, things turned around so much for the better when Stan took over and orchestrated the merge, which really brought the best of 2 worlds together. It brought people who were excited about being at the cutting edge of technology, together with people that did fundamental genetics and had this deep appreciation for the importance of graduate education. And so bringing those two those 2 worlds together, I think created a great department. So I'm glad to have been part of that early phase.
Yeah. So how did your research kind of change, or did it, over that time and with your transition to the Hutch? And did that have any influence on kind of what you were working on?
Yeah, it's I've not so much. I did become involved in a few more cancer-related projects, so I wasn't totally basic science, but it sort of continued on the same trend. I got involved in more computational biology areas because of people that were in my lab or people that I got to work with, but sort of followed the same themes still. But at the Hutch, I got to recruit new faculty, and that was.
So you had some new responsibilities and I guess roles. I think this is an interesting question about a publication story that you can tell us. Is there a publication kind of from your scientific history that has an interesting story to it, or controversy that?
She let me know that she was going to ask this question, so I did think about it. And I think it would be the publication about nuclear organization. So we had measured the distances between probes that were known distances on the DNA molecule for many probe pairs at varying distance, and we measured it for lots of nuclei. And so we had distributions of these distances and the relationship between the average distance and genomic separation. And the simplest model that came out of that could explain the data was that the DNA was acting like a polymer, a free-flowing polymer. No, nothing complicated, no fancy these genes holding on to each other or anything like that. It was just really biophysics. Mm-hmm. And so there were two, and I mentioned before that we use that information as a mapping tool. So, mm-hmm. So there are two aspects to that work that were kind of startling to me and made me think about how people view science. One was that when I got a grant from the what was then NHGRI, I don't know what it was called then, but I put in a proposal that I was going to use interface distances as a mapping tool, and this is my first grant, and I got this unbelievably good score. It was like 0.25 percentile. You know, it was just, how can it even? And then I was just waiting for the money to come, and it never came. and I thought, well, what should I? Well, maybe I should just call up. Well, it turns out that this was the early days, and the genome program at NIH had decided that there was already another group working on a similar problem, and they didn't need two. And so I'm
going to tell you?
I don't know. I think they were worried that, because I had people from the review panel, this was, a year and a half after the review, come up to me and say, how's it going? Because I had gotten such a, they remembered the grant proposal. And I said, well, they're not going to fund me. And so they had the problem of their reviewers being a little bit saying, well, why would we review for you if you trump us? And me thinking, I'm just this little starting scientist. How am I going to get into this? So I ended up having to plea to argue that just like they didn't have just a single lab in the country working on large insert cloning or genetic mapping, there needed to be some competition. So I had to say there should be two groups working on this, and then I had to explain what I thought was wrong about the other lab's approach, which was very distasteful to me, but it ended up working. So I got the money. So that was one. Then we ended up having this result about the polymer stuff, and sent it to Journal of Cell Biology and got the reviews back. And the review, they weren't going to accept it, and the reason was that one of the reviewers had said it didn't fit his preconceived notions of how the nucleus was organized. and there had to be more organization than just random, essentially. So I decided I wasn't going to just let it go. I wrote this response that was longer than the paper, in which I, basically argued that I basically said you can't reject papers that don't fit somebody's preconceived notion when there's no underlying data to support that notion, because then you will inflate or deflate the truth and inflate nonsense. But I didn't say it quite like that. So I got the paper published. So those are I picked those stories because they just were so informative for the world of science and how you sort of, not everybody thinks like you.
Right, right. And how to deal with that.
How to deal with it,
yeah. So I guess let's talk a little bit about how you think, not just your research, but kind of the field in general. So whether it's genetics, the technology along with that has kind of changed over the trajectory of your career, and what's been the most remarkable thing?
Yeah. Well, I'm sure some of you who are older than me find it even more remarkable. But I went to college when Sanger sequencing 100 base was a remarkable feat. I remember the biochemistry professor just being so excited about it, telling us about this. And now to think that you can sequence whole genomes for sense. So that's, I think, the most dramatic change. What really excites me is not the field that I worked in anymore, and that's why I'm retired. But the power of genomic analyses when applied to whole communities and ecosystems like oceanography or tumors, or where you can really examine the functioning of a whole community of organisms without actually necessarily knowing what the individual parts do, but you know that this community has the capability to fix nitrogen and do this kind of carbon fixation and that sort of thing. But so I think the real exciting phase is moving from that back to merging the worlds of metagenomics with single cell biology and fundamental biology, because now we really do need to know what functions are in a given organism, a given cell. It's like haplotyping from a population basis, but it's trying to understand what the individual organisms are capable of and how it works and how dynamic they are in an ecosystem. So I think ecology is going to be.
So you're coming full circle.
Yeah, and I want to be part of it. Yeah. So Heather knows that Gare, my husband, is now building flow cytometers that are going on boats, and he gets to go on these cruises and go work with oceanographers. So I'm trying to weasel my way into a lot of these trips as his assistant. And yeah, so that's one.
Have you been on some?
Yes. The most exciting one was off the north coast of Norway that probed up to the the edge of the ice, the Arctic ice, and sampled water all along where the Arctic and the Atlantic Oceans are merging, and and we were measuring phytoplankton, but there were people dredging up major organisms off the bottom. It was just really exciting. I hardly slept at all. It was just so that's an area. The other thing I think is where genetics and genome sciences really can what will be exciting to see what happens in the next decade is where environmental exposures and genetic risk merge, and given the fact that we can now sequence get the genetic information from lots of individuals and combine that with real measures of exposures, including food, and to try to tease out the relationship.
It's the real gene environment.
Yeah.
So you think we'll get there?
Somebody will, yes. Yeah, especially around, say the obesity epidemic and how that factors into disease. Yeah.
All right. Is there anything I haven't asked you that you want to talk about besides what you're doing now, which I'd love to hear a little bit about?
No, you've covered a lot.
So I hear you're still writing grants. Maybe you can tell us a little bit about what types of grants you're writing.
I couldn't believe that I got me. So I've moved full time up to our farm on the Skagit River, and so we have now about 300 acres that we're taking care of, forestry and farmland. Most of it rented out to other people, but we have animals. She's going to buy half of one of our pigs pretty soon, and chickens and rabbits. So if any of you want some good and sheep. So yesterday I was helping a neighbor with a difficult birth of one of her lambs. So there's still some biology and field biology involved. But what Heather's referring to is that I've gotten myself very interested in land conservation, and I'm now on the board of the Skagit Land Trust, one of the organizations in the Skagit Valley that is dedicated to protecting land for in perpetuity, either by buying it up or putting conservation easements on it. And as soon as I retired last May, I said, okay, use me. How can you use me? And then a month ago, I said, they're not actually using me quite enough. So I volunteered to help write a grant, which I can't believe I did because that's the thing I wanted to get away from. But this grant is for a whopping $20,000, and it's to help purchase and restore habitat in Barney Lake, which is one of the large wetland areas up near Mount Vernon, one of the last freshwater large areas there, big trumpeter swan area. So I'm getting back to little field biology, so I get to read about the rare frogs and the birds that it supports, and try to get some financial support to help make it happen.
Great. Yeah. I think we can probably open it up to questions if you're okay with that.
Bob, I was wondering if you had to fight for that $20,000 grant.
Well, when I realized that was their this is the reality. I mean, we really, even with the NIH being in such bad state, and the prospects for grants, we really have led a very lavish scientific lifestyle. They're not lavish, but we've been wealthy with the grants that we can secure for biomedical research, and then to go to an organization like the Land Trust or the Nature Conservancy and look at the portfolio of grants that they work very hard to get, and they are small, and they're going for every dollar that they can get, and they're smaller than a pilot grant would be in an institution like this.
And probably most people writing them are volunteers, really, like yourself.
Volunteers.
Right?
They've got some very good staff and biologists I can consult with, but it's a volunteer-driven organization. Yes, Steve.
Barb, back in the MBT days, were there projects that collectively a bunch of labs were working on together and brought together this idea of engineers and mathematicians and biologists, or did each lab kind of go off and do its own thing?
Yeah, that's a good question, because there wasn't quite the synergy that you might have, lab to lab. There was a lot of sharing of knowledge. But when I look back at my CV and ask how many publications did I have with Debbie or Lee Hood, and it's not a huge number. There were key ones where people brought technologies together. It was a question that I think Debbie might be better able to answer, because she was more in the middle of it with her sort of portfolio, and there were different personalities. I mean, I had some collaborations with Lee, but he sort of had his own, Lee Hood had his own thing going and had his own. So I had some, I did some fish for some things. We did Gary and I did an analytical paper about contig assemblies and things like that. So, and there were joint grants, things like that. But I think it was more bringing a lot of diverse people in that got excited about just being in this environment where they could go to, talk to Maynard and people in Maynard's lab, and talk to Debbie and then the biochemists that were around from Caltech.
So, Barb, chromosomes have been a persistent theme through a lot of your scientific career. And could you say a little bit about how your picture of chromosomes has evolved, and the way you think about chromosomes now as entities or organelles or embodiments? I don't know quite how to phrase the question, but I hope at least the gist of it.
Yeah, and
this is something you haven't been able to avoid. You've devoted a lot of your scientific career, and these have to have different meanings to you.
Yeah, interesting question. Yeah, because I'm not a cytogeneticist by training. I reminded Christine that I've hired people who could recognize the different chromosomes and tell me which one is being lit up. So I'm not a cytogeneticist, but um, how it's changed are from these stick solid X's to how it unfolds and moves in the nucleus. That's changed. And what I think is really cool now are the high C and 4C data that are really uncovering things that our polymer studies missed because we didn't have enough probes across the chromosomes. So we were looking at a more coarse feature that now people are finding areas in the nucleus that are more active and inactive, that there's movement when genes are called upon. So there is some functional stuff overriding with the more random biophysical unfolding of a chromosome at the end of mitosis. And I think it's so that's maybe the world, is that I don't think of a chromosome anymore as what a cytogeneticist would. I don't really care so much what the bands are. They were landmarks. I'm really more intrigued by what the chromosome does when it doesn't appear as a chromosome, that it's in the nucleus as one big mass of DNA and is exposed for use.
I don't know. There's a question in here, but it strikes me that you've had exposure to a lot of different ways that science is done. You were in Holland, and that's the European way, and you spent a reasonable number of years at
National Lab.
National Lab at a university at the Research Institute. And I wonder if you have any. Here's where I'm not sure what the question is. I'd like to hear you reflect on that a little bit.
Yeah.
What's good and what's bad, or what strikes you?
Yeah.
What works well, maybe.
Yeah. Well, I think what works best is hiring really good people and letting them do what they want to do, because and not trying to manage science. To find as a leader of a group of faculty is to just find ways to clear the path. I liken it to the guys that play that curling, that the scientists are that puck, and my job was just to sweep the ice so that puck would really go fast. So that's sort of part of the answer, but I was going to say something from the days in Holland that came into my mind that's gone now, but I think having colleagues who are not where there's an environment where you want criticism, and your colleagues are your first wave of asking for that criticism, and that there's a culture of accepting critique and giving it, and that's considered normal rather than sort of being on your own and trying to survive. And the best places for that were actually in Holland and the Hutch, where my last grant was successful because Harmeet Malik, who was a starting assistant professor, I asked him to read my grant, and he politely said, "Well, I'm not sure how to deliver this," but he just told me it wasn't good, and made suggestions about how to improve it. And I got the grant because he was not shy about telling me what was wrong. And in Holland, they had this once every 2 weeks, they had a evening seminar series in English, even if everybody was Dutch speaking there, preceded with dinner and beer, and it was tough. There would be people criticizing your use of a log scale. There were people, asking tough questions about why you had that as your control. But they did it in a way that it was about the science. It wasn't personalized. And in Dutch, you can shift to the formal you version of you and sort of distance yourself from it. But they had to do it in English, so they didn't have so when Gare first came to the States and Livermore Lab and asked some questions in those same veins, people came up and said, you've got to tone it down. You've got to be more tactful. You've got to figure out how to ask those questions in a more congenial way. Well, he felt he was being congenial because he was giving them criticism before they went out in the wide world and were exposed. His job was to help his colleagues. And as Dick Van Becken, who I mentioned at the beginning, the other thing that we learned from him, but I never could emulate it, was that he would stay awake until he had a tough question in his mind, and then he'd fall asleep for the rest of the talk. And so as a speaker, you knew exactly on what slide your question was going to come from, but I never been able to do that. Yeah. So I think those are the. Yeah.
You've been in many positions, from volunteer aide to division director. So you've seen lots of kinds of people, lots of different kinds of situations. If you were to give advice to this year's first-year students, what would you say?
I have more advice for a first year faculty member, but I suppose first year graduate student. Yeah, well, so follow your curiosity. It's tough. It's not easy. So there's I think Maynard said at one time where you're going up this career, and each time you're being thrown in with even more intelligent, more hardworking people than the last group you were surrounded. So you keep until at some point you hit the edge of your envelope, so realizing that a lot of it is self-driven, and you have to be tough because of that. To not expect other people to solve problems for you, that you'll get the most help from other people if you come with the problem you're facing. And this is not just science, but it's personnel management or grants or a collaboration gone sour, or all sorts of things. You'll get the most help from others if you come and not just say solve it, but that you come with a solution that somebody can work with. That you're taking a first step in solving the problem. And I think that's helped. The Hutch, my colleagues at the Hutch, there's not a whiner among them, and I think that's because they are that type of people. They're proactive and positive, and when they faced with something, they just don't throw up their hands and say, "This is somebody else's problem," but they try to help find the solution. There's probably a lot of other advice. No non-data ink and stuff like that. Okay, you're welcome. Thank you.